I am delighted to introduce Dr. Kristen Nowak. Dr. Nowak is an Australian Research Council Future Fellow at the Harry Perkins Institute of Medical Research and the University of Western Australia. She completed a Bachelor of Science in Biotechnology and a PhD in Neuromuscular Disorders. She has worked with Professor Nigel Lang since the late 1990s and co-discovered with him that mutations in the skeletal muscle actin gene, commonly known as ACTA1, is a cause of Nemaline myopathy. Dr. Nowak completed a postdoc with Professor Davies at Oxford University, pursuing whether cardiac actin could be a therapeutic target for ACTA1 disease. A strong focus of her current research is devising and evaluating potential therapeutic approaches for Nemaline myopathy, including using viral and gene therapy. And I had an additional slide that, oh, here we go. So without further delay, I will go ahead and switch over the screen view to Kristen Nowak. Are you ready, Dr. Nowak?
I certainly am. Thank you, Stacey.
Okay.
Great. I'll just hopefully be able to show you my screen now. Can everybody see my screen?
Yes, I can. Can everyone else?
That's great.
Yes.
Thanks, Rich.
Fabulous. Thank you. And I will try to keep my eye on whether there's any questions coming through, but Stacey has kindly said that she will keep an eye on that for me as well. But thank you so much, everybody. Good evening to you. It's morning for me here on the other side of the world in Western Australia. And thank you so much for taking the time to listen to my presentation today or tonight for you, and it may be in the future for people who will be listening to this taped recording. Please feel very welcome to call me Kristen, and as Stacey indicated, I'm really happy to take any questions along the way, because that means that we can hopefully address anything that I might not have covered properly, and I'll do my best to answer any of those. So I'll just hopefully be able to progress. I'll just give it this first slide as being a quick overview of what I'll hopefully talk about this evening. Many of you will know Nemaline, but I'll probably revert to the way I tend to say it here in Australia, Nemaline myopathy, very well, but I'll just give a quick overview as to what the disease, or the hallmarks of the disease are. Also, which genes can cause Nemaline myopathy. The cell and animal models that we have currently access to, which are really vital to our research. And then I spend most of the time looking at possible treatment approaches that have been used generically for muscle diseases, but also those that we've particularly been looking at applying to Nemaline myopathy. And then just give a quick conclusion slide. And then, as Stacey indicated, we can have another question session at the end and ask any other questions. And I'll have my email address at the end as well. And if you feel more comfortable emailing me afterwards rather than asking in this small open forum, you're very welcome to do so. In addition, if you think of anything down the track that you'd like to email me about, I'm very happy to receive your emails and do my best at trying to answer them. So what is Nemaline myopathy? And as I said, many of you will know this disease probably far more intimately than I do, even though I've been working on it, as Stacey said, with Nigel Lang since about 1997. So if we look at what the definitions of the words are, Nemaline means thread-like or fibrous, and myopathy obviously means muscle disease. So the disease, the hallmarks of the disease are these thread-like or fibrous aspects in the muscle biopsy. So it's a fairly rare disease affecting predominantly or exclusively the skeletal muscles, and these Nemaline bodies are what the pathologists will use to give a definitive diagnosis for this disease. It can range from being present at birth as a very severe form, and unfortunately patients can die within those first few days of life. But also we know that patients can have a milder or even an adult onset form of the disease that may be compatible with a normal lifespan. So we've got a whole spectrum of severities with this particular disease that often is thought to be a congenital disease present at birth. Almost all patients have weakened skeletal muscles, but we do know of a very small number of patients that rather than weak muscles, they've got very stiff muscles instead, and it's just a different process that's going on due to their defective genes. And this slide here, I'm showing two different ways of looking at muscle biopsies that have been taken from a patient with Nemaline. And on the left, we've got this Gamori trichrome stain, and what we're looking at here is a section through a muscle biopsy with the roughly circular shapes being the muscle fibres that are coming out of the page at you. And this particular stain is really one of the only ones that we can use on a muscle biopsy to show the dark blue structures here that we should hopefully see with the white arrows that are indicative of the Nemaline bodies. And on the right, we've got an electron microscopy picture called a micrograph, and the Nemaline bodies are these very dark, because they're electron dense, structures within muscle fibre, and these should not normally be there. So these two different pictures of what a pathologist and a clinician would use to give the definition of the disease Nemaline myopathy to a patient. So which genes can cause Nemaline myopathy? And you've probably heard of many of these. But first of all, I thought I should just recap why knowing the disease gene is so significant. And although someone can be given the name of their disease as Nemaline myopathy, having a particular genetic mutation can confirm that and give an accurate diagnosis. And in some cases, if someone else in the world has been identified with that same mutation, for example, and we've got these great databases that are available online that give the different mutations that have been found in those genes before, for many of the genes, the clinician may be able to offer a possible prognosis for the new individual that's just been identified as having Nemaline myopathy. And this can be really important because we know that for some very severely affected children, that if they get through that first period after birth where they are severely affected, that they might actually end up doing reasonably well after that time. So if we know that another person in the world has had that same prognosis or same disease course, then we can at least offer that information to new parents with a child with that same mutation. Obviously, genetic counselling can be better informed if we know exactly what the gene is that's causing the disease, and this is for diseases in general, and also whether the disease is being caused in a dominant or recessive fashion, whether only one bad copy of the gene is needed to cause the disease, or whether two bad copies are needed, so one copy is inherited from mum, one copy inherited for dad, for example, or whether the child that's got the Nemaline myopathy has got a new mutation, a new defect in their DNA that has not been inherited from their mum or dad. And the likelihood of having any further children with the disease can be determined or can be given an approximation based on this information. And then when a disease gene is known, families have the option to access prenatal and pre-implantation and even pre-symptomatic diagnosis. And pre-implantation, for example, is where a couple may undergo IVF and select to implant an embryo that has been screened for the disease gene that their family might be carrying. And as a researcher, knowing what the genetic cause of a disease is often some of the first important steps for us to devising therapies. So, because if we know that that's the main cause or the cause of the disease, we can go and look at trying to correct or overcome this particular defect to hopefully change the onset of the disease, prevent the disease, or lessen the severity of the disease. And on a wider scale, much of what we know today about muscle, for example, and this happens with other organs in the body as well, is through studying families with particular diseases that affect that organ. So studying families with muscle diseases have allowed scientists to actually discover new genes that are very important for muscle and for the normal function of muscle that were never known before until that researchers studied that family. So it's improved greatly the knowledge of the human body in general. So currently there are 10 Nemaline myopathy genes, and the early days it took a long time to discover what a new disease gene was. So the very first gene for Nemaline myopathy was identified as tropomyosin 3, and that was identified here in the West Australian lab of Professor Nigel Lang. And that took about 3 years to go from knowing where in the DNA code, where in the genome that gene was located, to actually identifying. But nowadays, we can, because of the different technologies for finding or for deciphering the DNA code, we have found many more genes far more rapidly. So here I've got popping up on the screen the 10 different genes that we know to date that cause Nemaline myopathy. And I've tried to indicate the size of the circle that they're contained within as representing how what proportion of the Nemaline myopathy patients that these disease genes cause. So Nemaline, Neb, and skeletal muscle actin, ACTA1, are the two genes that cause the majority of patients with Nemaline myopathy in patients around the world. And you can see that we have the other 8 genes that have different prevalences. And these gene names can be quite confusing, such as KLHL40 and 41, and they stand for different words and describing what the gene is. So the most common, as I mentioned, genes are Nemaline. It's a very large gene, and it has 183 parts or exons that make up this gene. And being large means it's difficult for us to screen as researchers and diagnosticians trying to identify whether a patient suspected of Nemaline myopathy indeed has a Nemaline mutation or a defect. But the other level of complexity is that this gene is very, very repetitive, and that adds another complication in our approaches to it, to be able to screen the gene to try to find the defect, or defects in this case, that cause the disease in the patient. So to date, all the errors or mutations in Nemaline are recessive, which means that the patient must have 2 defective copies to have the disease. So the Nemaline gene, we have 2 copies, 2 different chromosomes, or the same chromosomes that have this gene, but one chromosome is inherited from mum and one from dad in those cases. Sometimes mutations can arise. And we now know of hundreds of different mutations in this gene, so that's why it's a significant cause of Nemaline myopathy. Now, if we contrast that to skeletal muscle alpha-actin, or ACTA1, it is also a significant cause of Nemaline, but it's quite different to Nemaline in many ways. Fortunately for us, when we discovered that this gene can cause Nemaline myopathy, it is because it's a very small gene. It only has 7 parts or exons, which means it is much simpler for us to screen this gene. Although there is one aspect of the gene that is trickier to screen than others. It does prove to be a bit more of a problem, but we're aware of that, and so we can always tackle that slightly differently. So the majority of mutations in this gene are dominant, as opposed to recessive with Nemaline. And this means that a patient only needs 1 defective copy to cause the disease. But we do know that there are patients, about 10% of the mutations, patients with mutations in this gene have recessive Nemaline myopathy, where they have got 2 defective copies. So with the majority of patients having a dominant disease, I've skipped ahead there. So again, there's hundreds of different mutations in this gene, like with Nemaline, and because that most patients have a dominant disease when they've got actin disease, the mutations are de novo, which means they've arisen in that patient. The patient has not inherited the defective actin from their mum or their dad, because that would mean then that they would have the disease as well. But we know that most parents of children with actin disease don't have the disease, particularly for the severe cases. So it means that the egg or the sperm that created that child, the patient, that egg or sperm had a new mutation in actin, and that's why that child has got the disease. Are there any questions so far? If so, please feel very welcome to type them in or to unmute. But if not, I'll move ahead.
Hi.
Oh, sorry, yes.
Sorry, could you explain the last part? Because we just got the results for our test, the parental test, and my son is ACTA1. And we were negative for the blood, but the doctor said it's still 1% on the if it was a sperm or other thing. So could you just explain the last part really quick?
Sure, no, happy to do that. So that's right. So most patients who have Nemaline caused by actin mutations haven't inherited them from their mum or their dad. So the egg or the sperm that created them has got a new mutation in actin. And all of our cells, when they're dividing, whether they be egg or sperm cells or just other cells in our body, they often do get new errors in them because we've got 6 billion letters of DNA in our DNA code. So every time our cell divides and makes another copy of that 6 billion letters, our DNA machinery can introduce errors, just like a typist can introduce errors, even if they're an excellent typist. And we've got special correction machinery that does correct most of those, but sometimes one will slip through. So what can happen is when we have screened blood, so DNA that's extracted from blood of parents with a child with an actin mutation, the majority of the parents don't have a DNA or a mutation in their actin gene in their blood. But some parents do have a smaller proportion, and it might mean that they are a mosaic, which can mean that maybe a very small proportion. So when we're at a very early embryo stage, we start off as the one fertilised embryo, but then, or the fertilised egg, which is the embryo, and then we start dividing. And what we found with some families is that when one of the parents might have been at, say, an 8-cell stage in development, maybe one of those 8 cells has mutated and has got a new error in, for example, the actin gene. So then any cell that gets created from that one cell with a defect would also have the defect. But the other 7/8 of the cells that didn't have the defect, they will go on to produce cells that don't have the defect. So we have found that a very small proportion of the parents, or the couples, one of the parents does have a very small dose of the actin defect, and we might then go on and look at a skin biopsy or saliva and other sources of DNA from that person, and we can find a small proportion of the actin mutation, but we know that smaller proportion is not enough to cause disease in the parent, because often actin disease can be very severe and the parent obviously is not visibly affected. So that can happen, and also what can happen is the cell, say we talk about that 8-cell stage again, that one cell that might have created or had the error introduced, that cell might have gone on to produce the egg cells in the mother, or created the germline cells, the sperm cells for the father. So it may be that the chance of having another child with the same mutation could be as high as that parent having the full dose of the mutation themselves, because any other egg cell might have a chance of having the mutation, for example, or the sperm. But we know that this often doesn't happen, so the majority of cases will probably be that it was just that one egg that created the child with the disease, or the one sperm that had the mutation as well. So that might have been what your doctor was talking to you about in the case that there is still a chance that any future children could have the same mutation, because it could be that the mutation is present in your egg cells or your partner's sperm cells, for example, or it could mean that it was a one-off occasion. Does that help answer your question?
Yes, thank you so much.
You're very, very welcome. I'm sorry. Explain some of these concepts about pictures. I can assure you I'm using my hands a lot at this end, but you can't see those. Okay, so we'll go on to looking at nebulin and actin, for example, and some of the other genes or the proteins that these genes make, and what they're doing in the muscle cell. And what we've found is that all the genes so far that have been identified as causing Nemaline make a protein that is involved in the muscle thin filament, or make a protein that modulates or influences that thin filament. So in this picture here you can see that I've indicated where nebulin is and where actin is as well. And nebulin is a very long protein that makes up the whole the thin filament. And you can see that these little black rods, they're just little compartments of one big nebulin. Whereas the spheres, we've got 2 different types of colours here, they are all actins, but they're individual actins. So you could imagine actin being like a pearl on a pearl necklace, and the pearl necklace is made up of many actins. So nebulin is very, very big. You can see it's winding around many actins. I'll just go back here. So the thick filament interacts with what we call the thick filament in muscle to make muscles move or to contract. And I'll have a little video in a moment to just show that to you in a different way. Next point is just to reiterate what I was saying before, that we know that these proteins of the thin filament can cause Nemaline. And the nebulin protein, being so much bigger than the actin protein, it binds to as many as 200 individual actin proteins that are all strung on this pearl necklace. And in this diagram as well, just for your reference, these purple-shaped or spherical objects are the different troponins. So we know that troponin T, for example, can cause Nemaline. And the turquoise here is tropomyosin, and we know that tropomyosin 3 and tropomyosin 2 can cause Nemaline as well. And you can see that they're interacting with all these other proteins too. And so yes, nebulin is thought to be like a ruler to help determine the length of the thin filament, because it does span the entire length of the thin filament. Now I'll just show this little video, and I've got the source at the top there. I've shortened this a little bit, and this is just a little video from YouTube goes into detail about how muscles will contract. And as I'll just show you, unfortunately you won't be able to hear the audio, but I think that's okay because I'll give you a little bit of a voiceover. But the thick filament is at the top here, and what we've got down the bottom is the thick filament, and the thick filament is made up mainly of myosin. And you can see here that the troponin complex is being highlighted, and also tropomyosin and actin. But nebulin, unfortunately, is missing from this particular video, but you can imagine where it would be based on the figure that we were just looking at. The myosin heads down here, these purple heads of the protein, that binds to the actin in the thin filament, and you can see here that the muscle, that's what happens if they move past each other, the thick filament and the thin filament, when muscles contract. So I've wanted to just show you this so I can give you an impression of what might be happening, for example, in myopathy. If we have a defective actin protein, say this protein is no longer a lovely sphere, maybe it's changed shape a little bit, maybe it's a bit bigger or maybe it's a bit smaller, you can imagine how that will be influencing or potentially influencing these other proteins that it so beautifully binds to and interacts with, and the same with the thick filament. So maybe that may make muscle contraction or muscle strength be weaker. And we can imagine, for example, that the nebulin protein or the tropomyosin protein or the troponin protein, if these are missing or they're defective, how that could really influence and disrupt this very elegant process that happens to make our muscles contract. And if you wish to go to that source on YouTube, I hope you enjoyed watching that video. And just out of some interest here, what we do know is that many of the proteins, or at least some of the proteins that we know can cause Nemaline, can also cause other diseases. For example, myopathy, congenital fibre type disproportion, distal arthrogryposis, and distal myopathy. So these are other types of muscle diseases. But what's really interesting is that, for example, actin can cause many different types of pathologies in the muscle. So we know that actin mutations can cause Nemaline myopathy, but we also know that it can cause intranuclear rod myopathy or cap myopathy or minimal change myopathy. And a pathologist who would be looking down the microscope at a muscle biopsy taken from a patient would observe these different structures. And in some cases, and in fact probably in quite a few cases, would see that the patient has Nemaline bodies and maybe some of these other features as well, but would call the disease Nemaline myopathy. But other times they may only see, for example, intranuclear myopathy or intranuclear rods, and would call that disease in that patient intranuclear myopathy, or they may only see a fibre type disproportion. So I think it's really interesting that we know that we currently have 10 different genes that can all cause the same type of disease. They can all cause Nemaline myopathy. But we also know that some of these genes, when defective, can cause very different looking pathologies and can be called a very different disease. there is a thought that in fact some of these different pathologies are part of a spectrum. In fact, Nemaline bodies may not be so different to intranuclear rods or bodies and may also give us some other features. So that's something we still don't properly understand, whether it's just because of the time that we've taken that biopsy of a patient or the muscle that we've taken the biopsy from that we see different features in different people. So this diagram here just shows some of the different pathologies that can be caused by mutations in actin, for example, and these are different histological states of a muscle biopsy or electron microscopy pictures, and you can see that they look quite different even to a general eye, but for a pathologist many of the features in these different panels or different boxes would indicate that the patient would normally have a different type of label to their disease. But before we discovered the genetics of these diseases, we didn't know this. We thought that these are very different diseases. Before I move on to the cell and animal model section, are there any questions that you would like me to try to answer? Okay then, so we'll just go into cell and tissue models. And if you are a patient with Nemaline yourself or a family member of a patient, you would have probably come up against this question of whether you would be happy or happy for your child, for example, or family member, to donate some DNA that might have been taken from their blood or saliva, or whether some cells could be taken maybe from a skin biopsy or taken from the blood, or maybe the muscle biopsy that might have been taken to try to give a diagnosis.
Can anyone hear me?
Yes, I can hear you.
Oh, wow, you can? Oh, my son was diagnosed with Nemaline myopathy when he was about 4, but I don't know what gene it is. He was tested for the ACTA1, but that came back negative, but he's just low muscle tone all over.
Right.
But he's able to walk independently. And
Great. So yes, the diagnosis of the Nemaline myopathy would have been presumably from a muscle biopsy that was taken from your son. Yes, and in many cases it depends on the DNA or the diagnostic lab that the blood or the DNA would have been sent to. But in some laboratories will only screen, for example, actin as the first gene because it is so small and because it makes up a big proportion of the cases of Nemaline myopathy. So actin is a fairly easy one to screen and it gives us a good success rate. But then the next one, the next most obvious one, gene to screen would be Nemaline. But as I touched on before, it's technically very difficult to screen and there's lots of regions because if they're repetitive we can't screen very well. So some laboratories won't try to screen that gene, but others can. There are some places around the world that will try to screen that gene for you. For example, Dr. Karina Valgren-Pettersson from Finland does a diagnostic, well, it's more of a research diagnostic service looking at the Nemaline gene, for example, and there are various companies that will also look at it as well. What we, some other labs around the world, and our lab is doing that here in Perth, the diagnostic lab, they can have a gene panel, which means that all the Nemaline myopathy genes, for example, can be on this little panel, or this panel can be a small panel or it can be a bigger panel that will have genes that can cause all different types of muscle diseases, and then a patient's DNA can be screened using that panel, and then hopefully the gene that causes that disease in that patient will be identified from that panel. But what I should have said a little bit earlier as well is that although we know of 10 different Nemaline myopathy genes, there are still some people with Nemaline myopathy that are screened for those genes and don't get an answer as yet. And that tells us that there's at least one more Nemaline myopathy gene out there, or we're not quite looking in the right places of those genes to be able to identify all the causes of Nemaline myopathy for all the patients around the world. So it's possible you could talk to your doctor again and just ask if there's other places or other that might be able to screen some of those other genes for you and for your son's DNA. You're welcome to email me later if you like, and I can maybe give you some contacts, some people that I know will do screening for others.
Do you know Dr. Ian Glass out of Seattle?
I don't know personally, but I do know his name, yes.
Okay, I think he knows you. He said something to me about you a long time ago. Okay, and he tested Matthew for the ACTA1 gene. That came back negative, but he's got some of his DNA stored.
Yes,
Oh.
and for example, Dr. Alan Beggs, our great collaborator and friend in Boston, he does testing for other genes as well. And there's different laboratories in the US. There's different companies like GeneDx that will screen some of the Nemaline myopathy genes, and there's also places like us around the world that will screen other genes for you. So please feel welcome to give me an email, which I'll show you my address a little bit later on, and I'm happy to give you some of those contacts if you like.
Okay, I will. Thank you.
Great, thanks for your question. So yes, just to sort of touch on having a muscle biopsy is essential for to give a clinical diagnosis of Nemaline myopathy, and that's because that's the definition of this disease. But the DNA is essential for giving a molecular diagnosis. So as the question we just had was highlighting, you can still have Nemaline myopathy as the known cause of your known disease that you or your family member has, but you still might not know what the DNA cause is at that stage. And I was saying that we've been finding a lot more Nemaline myopathy genes over recent years, and there's still more or at least one more to find. Having a sample from a patient can be really crucial for us to be able to find these new genes, and often research laboratories such as ours will, when new genes are identified, we will go back and screen DNA samples that we have stored, providing we have got consent to do that, to see if any of the patients that we haven't been able to give a DNA diagnosis for have a mutation in any of the new genes as they become available. So the cells and samples are really important for us as well to test different possible therapies. And I know that some of you may have, for example, donated cells yourself or cells from your family members, and we've also got people on the line that work with these particular cell lines, and they're so important for us to be able to test some of our potential therapies in. And that's because they have the same genetic variation as the patient that they've come from. And we can also derive some of these cells from some of the animal models that I'll be talking to you about later and others, and we might be able to artificially engineer some of these cells as well. So they're a great first step for us to be able to test some of these potential therapies, but they're obviously a cell and they're not a whole organism, a whole animal or person. So we often find that having an animal model is very crucial as the next step to be able to test and evaluate some of the therapies that they're studying. So I'll just talk to you a little bit about the different types of animal models. They can be really essential for gaining more insight into the disease onset and progression. So if a muscle biopsy has been taken from a patient, that obviously has just come from one of their muscles at that one point in time. And we do know that some patients have multiple biopsies over time, but obviously this can be quite invasive and it's not normally something that a person needs to have. But if we have an animal model, we can look at different muscles from that animal and we can look at different times or ages of that animal to see how the disease might start and develop over time. And this can be really important. And the animal models are obviously a test bed that we can evaluate our therapies as well. And we can send animal models around the world so that different researchers can try their different approaches to therapies and can try to confirm results that have been found elsewhere. And so these animal models can be really useful to the whole international scientific community. For Nemaline myopathy, typically the models that we have are mouse models, and we have some zebrafish models as well. And there are also some fruit fly models too. However, to date, especially because many of or a good proportion of the Nemaline myopathy genes have been found only in recent years, there still are some of the Nemaline myopathy genes that don't have a corresponding animal model yet. But making animal models has become far less difficult over recent years because of some of the new gene technologies. So this next section of the talk, I'll just show you some of the different animal models that we have, and this one in particular we were able to make. I made this model when I was in Professor Kay Davies' laboratory in Oxford, and this is an Actin Nemaline myopathy model, and the numbers here, letters D286G, is the part of Actin that is defective in this mouse model. And like the Nemaline myopathy patients, these mice do have weak muscles. We've been able to study those. They have the Nemaline bodies, and you can see those in these pictures down the bottom. These electron-dense or dark features within the muscle that shouldn't be there. They also have some of the other pathological features that some Nemaline myopathy patients have. They weigh less than their normal mouse cousins, and we know that patients with Nemaline myopathy often have reduced muscle mass and body mass. And one of the models I made whilst in Professor Davies' lab was a model with this particular actin defect, but we also used the jellyfish green fluorescent protein that you may have seen before when jellyfish float underwater in the dark. Researchers have been able to isolate that protein, and now scientists can use that protein as a tool to help them with many different aspects in the laboratory. And one of the tools is to be able to essentially fuse or stick that green fluorescent protein to the protein that they're interested in to help track where that protein goes in the cell. We've been able to do that, and it's been really useful to see where the defective protein goes in muscle, for example. And one of the lines we made with this mutation is a much more severe than the others and has very early lethality. So we've got a severe form of this disease, and we've got a moderate form of this disease in the actin gene caused through the actin gene defect that we've introduced into this mouse. And this work here is the same mutation, a D286G mutation in actin, also using the green fluorescent protein from jellyfish as a tool, but this time it's not in mice, it's in zebrafish. And this is in collaboration with our long-term collaborator, Dr. Robert Bryson Richardson, who's in Melbourne, and he has made this fish, and it's very useful for us to be able to study the same mutation, the same gene, in two different species, in the mice and in the zebrafish. And you can see that where these arrows are, that we've got lots and lots of these brightly coloured rod-like structures, which are the Nemaline bodies. And Rob's also been able to test these fish for their muscle function and show that they have reduced muscle function. They can't swim as well as fish that don't have this mutant actin. So this is quite amazing, and it makes it possible, for example, with these zebrafish to test different drugs, for example, and then we can test them in the fish and then follow up with the mice. There's also an actin model which has the H40Y mutation, and this is one that was made by Professor Edna Hardman in Sydney, and we've also had the mouse here in Perth, and she's also sent them to many other researchers in the Nemaline myopathy field to do various studies on. And this particular mouse has got weakened muscles, like Nemaline myopathy patients. It does have Nemaline bodies and other pathological features, and you can see here the Nemaline bodies, and in this part B here, this is an intranuclear rod, which one of the patients who had this mutation was identified in a patient first. They also hit internuclear rods. But interestingly with this model and some of the work done by Dr. Mark Lawler has recently answered why this is, but 50 to 60% of the male mice die within 13 weeks of birth, but only a small proportion of the females do. This has been a really important model internationally as well for us to test different therapies for Nemaline myopathy. For Nemaline, there are also a small number of Nemaline mouse models that exist internationally. Most of these have a deleted Nemaline, so the Nemaline protein doesn't get made, and that can be very similar to patients with Nemaline myopathy caused by Nemaline. But because Nemaline is so important in muscle and other tissues, the mice die very early, within a week or two after birth. So it's a very severe form of the disease, which shows that Nemaline is essential for life. And one clever way of trying to overcome this is that some researchers have just removed the Nemaline from the skeletal muscles and not other tissues, and therefore the mice live, but they have the Nemaline myopathy in their muscles, and that allows more research to be done. Another mouse model for Nemaline is missing exon 55, or the equivalent of exon 55, which is section or part 55 of the gene. And this is a common defect in patients with Nemaline myopathy. So this has been made to model the same defect as the patients. And these also have a very severe form of the disease. They live less than a week. after birth. And there is also a zebrafish Nemaline model as well that exists, similar to how we were showing the Actin zebrafish model just in the previous slides. The Nemaline zebrafish model does have Nemaline buoyance, and it has reduced muscle strength as well, and also dies quite early. And our laboratory now here, thanks to funding from the Foundation Building Strength for Nemaline Myopathy, has been studying some new nebula mouse models with different types of mutations. And we are really excited that it looks like these new mouse models are going to be very useful for helping us try to understand the disease process in Nemaline Myopathy due to nebula mutations, but also to help us test some of the new approaches that we're investigating for hopeful therapy or treatment of Nemaline Myopathy due to nebula mutations. Before we get started on the next section, are there any questions that you'd like me to try to answer about animal models or cell models from patients?
I have a question really quick. Another one, sorry.
Sure. That's okay.
Our son was diagnosed with the ACTA1 and it's G247R. I don't know if you have any information about that.
Off the top of my head, I don't have information about that, but I help with Professor Nigel Lang look after the database of the different Actin mutations, so I can certainly look in there for you and see whether it's ever been identified anyone else in the world. I do know, though, for example, that there isn't a model for that particular mutation as yet. There's still only a small handful of mouse models and zebrafish models for Actin mutations. It often takes us a long time, and it's been very expensive to make mouse models and indeed zebrafish models as well. But the new technologies for changing DNA, which I'll talk to you about in a little while, not only are they useful for us to try to treat DNA defects, but they're also very useful for us to try to make new models. So we basically introduce the DNA defect into a mouse or a fish, for example. So in the last 2 or 3 years, making new models has become faster and less expensive than it has been in the past. So the hope is that more animal models for the different diseases that people are interested in around the world will be able to be made more efficiently. So it's possible that models for more Actin mutations and more nebula mutations may be done or created in the future. But it's really important as well that we can test certain approaches in cells from patients. So, for example, some of the technologies we're trialling, we're hoping that we can trial those in cells. It might be skin cells, it might be blood cells from patients with different mutations and different Nemaline Myopathy genes to see whether our approaches work for more than one of the mutations. So that's something that we could, or you may be able to consider for your son as well. But again, I'm happy to tell you a little bit more about that mutation later. I can look it up for you and see if there's any other information that's known.
Oh, that would be great. Thank you so much. I will send you an email later then.
Yes, no troubles. You're very welcome. So if we look at treatment approaches, and I guess that's where the majority of us are focused when we're studying Nemaline Myopathy, even if we're trying to just, well, it's not just, but even if we're trying to understand the disease process, that is because we're not just simply interested in that, it's because we're hoping that information will help us and guide us into devising better treatment approaches. So we ultimately want to be able to develop a therapy or a prevention for Nemaline Myopathy. And what we have done is looked at the different approaches that are being used for skeletal muscle diseases in general. And unfortunately, skeletal muscle is a really difficult tissue to target because it can make up so much of a person's body. It can make up to 40% of a person's body mass, and it's obviously very widespread, quite different, for example, to just treating an organ, an isolated organ such as an eye or maybe a kidney. So it does have that technical challenge, and delivering into muscle has been difficult over many years. But what I'm going to do in this slide is just show you some different approaches that people have been studying over the last 2 or 3 decades for skeletal muscle diseases. And this is essentially what we did when we, for example, discovered the actin mutations, we thought this is fabulous information, and it's obviously really important to the families and the patients that they know this information, what's causing the disease. But now that we have that information, how can we use that to try to develop therapies? So what we did was look at approaches around the world that were being used for other muscle diseases, and most of those muscle diseases were the muscular dystrophies because they are more common muscle diseases, so more effort has been put towards them over the years. So one of the obvious approaches is gene replacement therapy, and this is when the disease is caused because through the absence of a particular protein or the absence of a normal gene that's functioning. And for example, Duchenne muscular dystrophy is caused by an absence of the dystrophin protein. And so a lot of attention has been used, or has been focused towards this type of approach. So to replace a missing gene or put in a functional copy of a gene, there's different ways that people have looked at that. And one of those is using a DNA plasmid, which is not found in the human body, but it's a small, basically a DNA circle where you can put in the gene, include the gene, and you can try to deliver that to the muscles, for example. And that has had some efficiency, but hasn't been progressed more recently. Viruses can be used. We know that viruses can be really nasty in some instances, but they can be very useful as a research tool and as a therapy tool, where some of the nasty genes can be removed, and they make space then for the genes that we want the viruses to deliver to the body, so that we can put those genes in and then use viruses that we know have a preference for targeting skeletal muscles, for example, and we can get those viruses to do some great work for us. They can deliver the missing gene to the muscles of the body. And so this therapy, even though it has traditionally sounded quite scary, using a virus, but this approach has taken off over the years and has had some great successes. And also another way to deliver a missing gene is to deliver that inside a cell, either a myoblast, a muscle cell, or maybe even a stem cell. And people have shown, for example, that fat cells can be converted into muscle and can be used to deliver some muscle cells to areas of the body where the muscle has been degrading. So these different lines of inquiry have had different successes, and sometimes what can happen is a cell, like a muscle cell, can be taken from a patient and then can be grown in the laboratory in tissue culture, and that patient cell can be, or the cells, because they will divide and create lots of cells, they can be corrected for the mutation, or perhaps the missing gene or defective gene can be replaced with a good copy, and then it can be put back inside the patient. Or patients from a donor person can be used, but then you might have instances of immune response and rejection because that cell has come from a different person. You may have heard of aminoglycoside antibiotics, and these antibiotics can read through the signal that DNA has for the gene to be stopped, or when the RNA and protein is made from that gene. And scientists have found out that these antibiotics can read through a premature stop variant or error. Some in different genes, and we know in the Myopathy genes, some of the errors are causing an early stop. And so these particular drugs can be used to get the machinery of the cell to read through that. So basically ignore that stop signal and keep going to the real stop of the gene. To my knowledge, no one's used these drugs for the Nemaline genes, but they have been used for dystrophin gene, for example, and some other genes that cause muscle diseases. This next point is trying to correct the mutation, the DNA error. So this is perhaps the most obvious way, as well as of trying to treat a disease, so that if we know that this particular DNA error is causing the disease, then how can we correct that and overcome that? And until the last 2 or 3 years, the technical ways of trying to do this have been quite limited and have presented hurdles. So although people have been trying to do this, the technology just wasn't available for this to be very efficient. And although they could maybe do it in reasonably successfully in some patient cells in the laboratory when they tried to treat, for example, a mouse. This was very inefficient. But I'll go on and touch on this point again in some of the slides that are coming up.
Dr. Nowak, there's a question from the audience.
Yes.
She's asking, would a cell from a full sibling be a good donor?
Right, so from the, I guess it's very similar to donating a bone marrow, for example, or an organ. It's very possible that that sibling, well, there's two siblings should have about 50% of their DNA genome in common. So they would have much closer similarity to a cousin or, in fact, someone that's not related. A parent should also have 50% similarity with their children as well. So a parent or a sibling should have about the same amount of similarity. But yes, I guess from an immune response, a full sibling would be very useful, or perhaps more useful than a complete stranger who was donating a cell from that point of view. I must say that I'm not a clinician by training, I'm a scientist by training, so my answers are coming from that perspective. But in terms of just the general idea of using an organ or for a replacement or a donation, usually a family member has a much greater chance of being a better match. So if we go back to the antisense oligonucleotides, it's a bit of a mouthful, but this particular approach has shown some great success over the years, and particularly recently, and I'll show you a slide in a moment, for Duchenne muscular dystrophy. And this technology was pioneered here in Perth in Western Australia by Professors Steve Wilton and Sue Fletcher, who have been colleagues of ours. And I'll show you that in a moment, a little bit more detail. And upregulation therapy is one of the approaches that we have tried for actin disease. And upregulation therapy means that you use another gene and another protein, hopefully from the same protein family or gene family. Most genes and proteins in our bodies belong to a family where the other genes and proteins in that family are very similar. And you can use another gene in that family to hopefully compensate and overcome the defective gene or protein that a patient has. Sometimes, though, it can be another gene or another protein from the same pathway in the body, but not the same family. And this is what we've been trying to do by using a different actin, a heart actin, to help try to treat Nemaline myopathy, which is caused by defects in the actin that's in our skeletal muscles. And it might be counterintuitive, but exercise has also been looked at as a potential therapeutic for different types of muscle diseases. And for some muscle diseases, it seems that moderate or low impact exercise can be beneficial. High impact exercise can obviously destroy muscle and break down muscle, and if muscle can't regenerate properly, or obviously regeneration has a certain limit, this is not good. But sometimes low or moderate exercise can be has been shown to be beneficial. And another aspect that I won't go into too much detail here, but you're very welcome to look up and you'll find some information online about this technology, is RNA interference. And this technology is where you can try to target one bad copy of a gene for silencing, and just keep the good copy of a gene active. And that can be very useful for dominant disease where a patient's got one good copy and one bad copy. We'll go into a similar principle in a moment, but I won't say any more now in the interest of time about RNA interference. So antisense oligonucleotide exon skipping is what I was touching on before, and this is where a gene's got many parts, and this technology can skip over a part or an exon of a gene that has the defect. And this has been really successful for Duchenne muscular dystrophy, as I mentioned, and also survival motor neuron gene for the disease spinal muscular atrophy. And Duchenne muscular dystrophy gene, dystrophin, is a very large gene, and we know that different parts of the gene can be skipped over or not included, and the protein that's made can still be quite functional. And this is the technology that's been used. And this is Steve and Sue that I mentioned to you before, and just recently in September, the FDA approved one of their drug designs for treatment of Duchenne muscular dystrophy boys. And so this was a really significant step forward, and they have got other designs for the other defects in this gene as well. To my knowledge, no one's used this approach for the Nemaline myopathy genes. It's probably because we really don't know enough about these genes yet, especially big ones, Nemaline, that this might be applicable to. We don't know yet whether any bits of the gene can be skipped out and still form a functional Nemaline. But we do know, for example, for actin, which is a very small gene, that we can't just skip a little bit out of it and it still be functional. We know that that's detrimental. So this approach would only be applicable to some of the Nemaline myopathy genes, maybe tropomyosin, for example, because we know that tropomyosin has different parts of it that can be alternated with other parts. So that is a potential approach. And that just reiterates that there. So upregulation of an alternative gene, the laboratory that I went to work in Oxford, from Professor Kay Davies, she has pioneered looking at eutrophin for Duchenne muscular dystrophy, for the absence of dystrophin. And so eutrophin is a fetal version of dystrophin. And we know that in the body, when we're fetuses inside the womb, for example, that we have a certain set of genes that are switched on. And often around birth, these genes get switched off and other genes from the same gene family get switched on instead. So we can have fetal genes and we can have adult genes. And we presume that this occurs because of slightly different functions of these genes. So we know that cardiac actin, the actin gene that's in our hearts, which is making our hearts beat now for all of us, is also a fetal version of actin in skeletal muscle actin. I'm sorry, it's a fetal version of actin in skeletal muscles. And this is some of the work that Kay's been working on. She's been working on eutrophin over the last 2 or 3 decades, and they're now in phase 2 trials for their drug that switches on eutrophin after birth when it's normally not switched on. So our question was, could we use cardiac actin, like Kay has been using eutrophin, could we use that to treat recessive actin Nemaline myopathy? So recessive being due to 2 bad copies of actin, and that's for about 10% of the patients. So cardiac and skeletal muscle actin are 99% identical, which is just amazing. We couldn't have hoped for a closer match between 2 different proteins if we're trying to get one to substitute for another. And as I touched on just before, that it's the major actin in our heart, and that's why it's called cardiac actin. But it's switched on in our skeletal muscles when we're fetuses in the womb, and it's the main actin during this time. So we know that this protein can do a good job and can function well in skeletal muscles during development. But around about birth, it was known that it was switched off. And that's when the skeletal muscle actin, or the ACTA1 gene, takes the lead role. So the good thing about this concept was that if it worked, if we can, or if we could have substituted cardiac actin for skeletal muscle actin, it's possible that it's more of a one-size-fits-all or one-size-fits-most approach for treatment, rather than correcting individual mutations, for example. So we studied mice that had no skeletal muscle actin in their muscles, like some of the Nemaline myopathy patients. So they were defective, they had 2 bad copies of this gene. And normally these mice all die within 9 days of birth or after birth. So it's a very severe model of Nemaline myopathy. And what we did is we artificially turned on the cardiac actin gene in their skeletal muscles after birth. So normally it's switched off at that time, but we artificially turned it on with a technology that we can use for mice. And then we found that these mice lived until over 2 years of age, which is old age in mice, and they functioned really well. And this is Nigel Lang and myself holding one of these mice is the treated mouse, and the other one is its brother that was effectively a normal mouse. And here we are on the first birthday of these mice, which was a significant step because we were able to show that the heart actin could effectively replace the missing skeletal muscle actin. And these the pictures just show on the left-hand side the control mouse, muscle from the control mouse, stained with 2 different stains, and these are the rescued mice. And you can see that there's none of these big aggregates that indicate Nemaline bodies, and the muscle looks quite normal. So this showed us that the heart actin could substitute well for the missing skeletal muscle actin. So that was a significant step in the right direction. It was what we call a proof of concept experiment. We showed that in mice this approach could work. And we also have gone on to look at the dominant actin nemaline using our mouse model with a D286 gene mutation. And in this case, we think that when there's dominant disease, the bad copy of that gene is maybe a poison copy or a poison protein, and that's what's causing the disease. So we weren't sure whether putting the cardiac actin into this mouse model would be beneficial or not. But we found that by doing this, we were able to significantly improve the lifespan of the mice, even though they did still die earlier than the normal mice. We improved their reduced body weight that they normally had, and they were able to, we put them on little running wheels, and they were able to run a normal sort of distance compared to like normal mice do. But the mice still had some of the muscle pathologies that we see in these mouse models. So, but this was very promising that we could at least decrease the severity of the disease in this dominant actin mouse model of Nemaline myopathy. We also tried using this approach on the H40Y mouse model that I mentioned earlier, but unfortunately we couldn't rescue these mice. They still had the same severity of the disease, and it's probably too complicated to get into here, but we think there's some reasons why this might be. But one of those would be that perhaps this approach is more mutation specific than we thought. Perhaps some of the defects are more poisonous than others, and we can't dilute them sufficiently. But we can always talk about that more later. So the problem with this technology is that what we did, the way that we could increase cardiac actin was through something called transgenesis, which we can do with mice, but we're not allowed to do with humans, understandably. So what we have to do now, what we've been spending many years on, is how can we increase the levels of cardiac actin in muscle after birth in an approach that would be able to be applied to humans. Can we get a drug to do it? Can we get a drug to stop cardiac actin being turned off after birth in muscles? Can we get a virus to deliver the cardiac actin? And can we somehow genetically engineer it in a way that can be applied to humans? And some of these approaches is what we've been doing with our research project that's funded from a foundation Building Strength. Stacey, I realise that I'm going far more over time than I was expecting. Are you happy for me to keep going?
I am very happy if you are.
I am, and I guess
Okay.
if people need to leave it, of course, that's understandable, and you've always got the option to listen to more of the talk at a later time from the taped version.
Exactly. Thank you so much.
Right. No, you're very welcome. So was that another question that I can now see on the right from May about the... maybe I can get to that one if I've missed the time. I will get to that during the question section. So this here, CRISPR-Cas9, it's a bit of a mouthful, and it stands for a bigger phrase, but it's something I was hoping to talk to you about because it's a really exciting development or set of developments that's been happening in the last 2 or 3 years internationally. And you may have even heard about this in the popular scientific press because it's touted as this amazing technology that will be very, oh, thanks, mate, I just saw your message, it will be hopefully sort of a holy grail in terms of treating a whole range of different human diseases. So I will show you in a few slides' time a couple of links to some YouTube videos which I recommend will explain this technology in more detail for you, but I've basically tried to summarise it here in a somewhat non-technical way. But what this CRISPR-Cas9 technology can do is it allows DNA sequences or genomes, which is our full set of DNA, to be edited with unprecedented precision, efficiency and flexibility. And this is what makes it so amazing. And also that it can be done fairly easily without any specialised equipment. So this system comes from bacteria, and essentially it's the bacterial defence system or their immunity against viruses. And the very clever researchers who discovered this system had the knowledge, or they had the foresight to predict that it could become very extremely useful tools for making new models of disease, for example, for better understanding different genes and how they function, but also for trying to treat diseases. So the Cas9 is an enzyme that can snip DNA, and CRISPR is a collection of DNA sequences that can direct the DNA, or sorry, can direct the Cas9 where to step. So we can direct this particular enzyme to a place in the genome that we're interested in, and then ask it, well, don't ask it, we take it to that part of the genome and it can snip and cut the DNA. So our cells will try to repair DNA after it's damaged, and this repair can help knock out genes or disrupt genes if we're trying to understand what those genes do. We can also insert a reporter gene. So I touched on the green fluorescent protein from jellyfish. We can try to insert that gene into the location of a gene that we're trying to work out what it does, and we can then see where that green fluorescent protein is expressed. It can also insert DNA mutations, and this is the technology I was touched on earlier that is helping speed up and also lessen the cost of making new models of disease, because we can insert mutations into selected areas of the genome and in fact multiple regions at the same time. But what we're really keen on pursuing further is how we can use this technology to correct DNA mutations for Nemaline. And these two links here are some videos. There's many videos on the web now that try to describe what this technology does and how it does it. But these are two that I recommend that you may wish to watch if you want to know this in more detail. And another way of using this technology, which is really taking it to another level, is that it's a little bit more complicated. So I'll just try to explain it the best I can in a short period of time. But the Cas9 protein can also be stitched together with other proteins, and so that you make a protein complex. And what can happen is that the Cas9 protein can also be engineered so that it no longer cuts DNA. So what it does, though, is it recruits these other proteins to an area of the DNA, of the genome. And what we can do is we can target that Cas9 and the proteins that it's joined to go to a promoter. And a promoter is the area of a gene that switches that gene on, and it's the region that can be silenced and switched to turn that gene off or down. So what we can do is artificially switch on or artificially turn down how much that gene is being expressed, that gene's being made. And people are looking at this, for example, in cancer to try to change the expression of different genes. And what we're wanting to do is to try to get this technology to switch on cardiac actin in the cells and mice so that it's switched on in a time that it's not really switched on. And we're hoping that this is going to be successful. So earlier this year, in a very prestigious journal called Science, there were 3 papers that were published using this technology, the CRISPR-Cas9 technology, to treat Duchenne muscular dystrophy. And these are really exciting papers and very significant in a step forward for using this technology to treat muscle diseases. The Duchenne muscular dystrophy gene in the mouse model has got a particular type of mutation that's not a common type of mutation for the Nemaline gene. So it's going to be more difficult for us to use this technology for Nemaline. But it's a very significant step forward, showing that you can treat the eye muscle disease using this technology, and they were able to deliver it, for example, using viruses, either very soon after birth or a little bit later after birth, and get great success and delay and essentially prevent the disease from being from onsetting. So this is a really exciting technology. So looking at CRISPR-Cas9 for Nemaline, we may be able to use it to stitch together parts of genes. And it might be useful, for example, for tropomyosin, again, where I mentioned to you previously that it has different parts of the gene that sometimes are stitched in and sometimes stitched out, depending on whether it's the tropomyosin 2 gene or the tropomyosin 3 gene or even the tropomyosin 1 or 4 genes. So that might be applicable, but it's not necessarily going to be applicable for the other genes of Nemaline. But what we're hoping, one of the other applications, is to correct the individual mutations, as I touched on before. So we've been using this approach in our research projects that are again funded by their foundation Building Strength to look at specific mutations in the ACTA1 gene to see whether this technology can correct those mutations, and also looking to see whether this technology can correct nebular mutations. And essentially, this technology could be used to correct mutations in other Nemaline myopathy genes too. And as I indicated with the other application of this technology, we can use it, the CRISPR-Cas9, to upregulate or switch on alternative genes that might compensate for the ones that are defective. And we know about cardiac-actin for skeletal muscle actin or ACTA1 nemalinopathy. But unfortunately, most of the other nemalinopathy genes don't have a fetal version. For example, Nemaline doesn't have a fetal version that we're aware of, so we can't use this technology to try to modulate the expression of that. But hopefully I've shown you here that there's different applications from the same technology that can be applied to the different nemalinopathy genes. Excuse me. The CRISPR-Cas9 technology does have some pros and cons. So one of the cons perhaps, and I see it more just as a hurdle that we just have to make sure that we can eventually jump over, is that we need it to deliver to skeletal muscles. So we're likely, and this is what we have been doing in our research projects, going to use viruses to deliver these particular tools to muscles. Viruses have different size capacity limitations, but that's something that we're able to try to engineer over. And it's possible that we can put in special promoters to switch on the Cas9 technology only in muscle, for example. But the hope is that this technology using viruses may mean that it can be very long-lasting, which could be seen as a pro, but perhaps could be seen as a con as well. And we just don't know well enough yet, if we're trying to treat a mouse model or even a human with this technology, whether we might have what they call off-target effects. Is this technology going to also, by accident, target a gene that we don't want it to target? And maybe accident's the wrong word. Maybe it's doing what it's supposed to do, but we just didn't foresee that. Another way of delivering the CRISPR-Cas9 machinery might be using nanoparticles, which are very small particles that can be targeted to different parts of the body, for example. And another downside, which is getting a little bit technical here, but there are different ways that the cell can correct DNA once it's been cut. And the way that we need it to correct is called homologous directed repair, HDR, if we want to try to correct a mutation. But this is not the preferred mechanism of the cell. It only occurs in dividing cells. And our muscles don't divide for very long after birth. They stop dividing quite soon after birth. So these have been some challenges that we've been looking at in our research project. But what's really exciting is that there are new technologies, there are new advances on the CRISPR-Cas9 approaches that are happening all the time. And even last night I saw a paper that had just been published yesterday in the Science Journal, which was coming up with a different version of this technology that may be able to overcome some of these hurdles. So I'm very hopeful that although there are hurdles, we will quickly or soon in the near future be able to overcome some of these. Okay, so to try to summarise very briefly what I've been talking about in this presentation, there are still hurdles to overcome, but hopefully you can also feel that the hope that us in the research field do, that the technology is advancing at an incredible rate. Gene sequencing technology has advanced over recent years, and that's why we now know of more nemalinopathy genes than we did before. And that's why we're hopeful that this technology will help us find at least one remaining nemalinopathy gene that must be out there. This CRISPR-Cas9 gene therapy technology is super exciting, and there, as I touched on, are regular updates and advances all the time. And viral delivery has been progressing over recent years and decades, and this is becoming more and more applicable and safe and useful for different types of muscle diseases and indeed diseases in general. As I hopefully demonstrated, knowing the causative gene is often the first step for researchers to be able to develop therapies and to test them and to have that rational design. And also using cell models or animal models are crucial for us to be able to test these approaches. I've just touched on today in this talk some of the approaches that we've been using in our laboratory, and we are using more than these approaches as well. And we know we're very fortunate to have a strong network of collaborators and researchers around the world who research nemalinopathy, and some of them are using similar approaches and some are using different approaches. Some of them are using our mice with different approaches, for example, and so we know that there is a multi-pronged approach being attempted to come up with successful therapies for Nemaline myopathy. Our hope is that various therapeutic approaches might be synergistic so that we might be able to get some benefit, maybe 10 or 20% benefit from one approach. We can combine that with another approach that might give us another 10 or 20% benefit and so on. And so we can come up with a combination of approaches that might then be very effective. And what is reassuring is that there are many researchers working on Nemaline myopathy, but there are even more researchers working on muscle diseases in general, and that we know that advances that can be made in the muscular dystrophies and other types of muscle diseases are most likely able to be applied to treating Nemaline myopathy, and we just have to stay very aware of those and try to translate them into the genes that we know cause Nemaline myopathy. So to finish up with some thank yous, I really appreciate you listening to me here this evening and asking questions, and hopefully you'll ask me some more soon in a moment. But we're really grateful to you as patients and families for supplying cells and your DNA for us to be able to study. We're very grateful for all our wonderful collaborators and colleagues around the world, and very importantly the funding bodies that allow and facilitate us to do this research. And I listed here the Foundation Building Strength for Nemaline myopathy, and for other research foundations and institutions that have funded us over the years that have made all this work possible. And unfortunately it is a very long-winded process when you're making these animal models and trying to test them, but we're hopefully making significant steps along the way. So thank you very much, and I'm now very happy to take any questions. That I appreciate that I have gone over time, and that it's evening time for you, so if you prefer to take down my email address and email me at your convenience with questions, I'll be very happy to try to answer those when they come in. But otherwise, please fire away and ask me any questions now, should you wish.
Thank you so much, Kristen. I really appreciate you doing this for us. I have a question that I would like to start with, and I think that May's question kind of gets at what sort of an overarching kind of question that encompasses a lot of things. But what I would like to hear is, do you have any ideas that you can speak to that would tell some of the families who have the Nemaline gene affected in their families how some of these approaches might be applied to that very large gene?
Yes, I'm happy to try to do that. So with Nemaline, we have been looking in the laboratory at trying to correct some different Nemaline mutations, and we've focused in on a couple of mutations that our new mouse models have, purely from the point of view that if we can try to correct those in tissue culture, we then have a mouse model that we can work, then test the next steps with. But yes, being able to, with Nemaline, there are 2 defective copies that patients have. So we know though that the parents, in most cases, carry one of those mutations, and a patient might have 2 identical mutations. They might have the same one that's inherited from mum and dad, and this often happens, for example, when the exon 55 is deleted. But we also know that many patients have 2 different mutations in different parts of the Nemaline gene. But we know that the parents who carry these gene defects or have 1 copy are not affected. So I guess our approach is, can we change at least one of the mutations or correct one of the copies of the gene, the Nemaline gene, so that we can convert a patient with Nemaline myopathy into something more similar to their parents, which are not affected. So can we get something even in between that decreases the severity? So we've been looking at trying to correct those mutations, and we've had some success, but I've touched on briefly some of the technical hurdles that we've had, that the approach that we've been applying to correct a mutation needs a particular type of repair, and that's been difficult. But there have been some advances recently that indicate we might be able to sidestep that approach instead. But what we're hoping, for example, with the exon 55 deletion is, can we put that exon back in? And it's quite a big deletion that happens in patients that encompasses exon 55, which is only small. It's only 105 nucleotides or DNA letters long. Can we put that exon back into the gene and have some sequence either side, some DNA either side, rather than replace the whole big section, which is over 2 kilobases large, I think. So we've been trying that, and the technology that I mentioned was published yesterday gives us another idea as to how we might be able to do that and do that far more efficiently. So I'm happy to expand on any of that if you like. Stacey, has that somewhat answered what you were hoping me to, or to address what you were hoping me to?
Yes, I think that's fine. There are a number of families that I know of who do have the Nemaline gene affected in their families, and I just think that they were hoping to hear, you know, how the technology could possibly be applicable to their families. So thank you for stating that.
Yes. You're very welcome. And I do have some slides that I could try to explain that more visually, and we can always perhaps do that on a separate webinar, or maybe I can make those slides Stacey, and she can put them on the web, and I can just have some little points underneath and just a diagram of showing how we do that. But essentially with the exon 55 deletion, we're going to put in that missing exon 55. Can we put that in? And as I spoke about earlier, the technology, for example, can put in a green fluorescent protein, so why can't we put back in exon 55 instead? We've also been using this technology to try to correct the actin mutations. And technically, that's probably going to be a little bit easier for us because most patients just have one defective actin and one actin gene that's still normal. So we're only having to target the one defective copy. Whereas with Nemaline, we've got 2 defective copies. But as I was saying, we know that the parents normally of children with Nemaline due to Nemaline, they also have one defective copy and they don't have the disease. So we're hopeful that we can just target one of those copies and still get a positive outcome in terms of the disease presentation.
Great. Would you like to take May's question next?
Yes, thank you. So thanks for that, May. And I'm sorry I didn't see it when it popped up. Has she had to leave the... Oh, no, you're still there, May.
I'm still here. Hi.
Hi. Thank you. So yeah, the cell samples are, DNA samples are really important for being able to give a molecular diagnosis, a DNA diagnosis. And because essentially we can't give a diagnosis at a DNA level unless we can study DNA. So this is important, especially in the future, because if we do devise a therapy that is able to be translated into the human patients, we need to know what the gene defect is first. So, for example, if we are successful at getting this CRISPR-Cas9 technology to correct actin mutations for Nemaline, if a patient's got Nemaline caused by Nemaline, a Nemaline mutation, then that approach is not going to work. We need to know that they have a Nemaline defect or defects and which ones they are. So it's really important from that point of view, and there's a big push around the world for especially patients with rarer diseases to have their DNA diagnosis identified if possible and to become part of registries as well, because the hope is that if treatments become available and particularly say if clinical trials become a viable option, that patients know what their gene defect is. So that's one thing I can mention. And also for us to be able to discover new disease genes, we need to, for Nemaline, we need to have DNA samples from patients with Nemaline to do that and to be able to screen the genes we do know that cause the disease, and if we can't find an answer in those, then look and see what else we can find. So that's really important. From the point of view of a sample that might be some cells, maybe a little bit of skin sample or blood cells, for example, or even some muscle cells taken from a small muscle biopsy, they're going to become more and more important, I think, in terms of us being able to trial these treatments. And for some of the treatments that we've been looking at over the years, we could only really test them in an animal model, but the CRISPR-Cas9 technology can be tested in tissue culture, and we know that it's fairly easy for us to design different, how would I say, models of the CRISPR-Cas9 technology or versions of that technology for all the different mutations that exist, for example, in Nemaline and in actin. They're fairly inexpensive in the big scheme of things to be able to do, and fairly easy to design and get made. But we know that we won't be able to make animal models for all those mutations. There's over 200 mutations in actin, and there's a similar amount in Nemaline. We can't possibly make 200 different mouse models for actin disease. But we can test those different mutations in tissue culture. So I think it's going to become more and more important for us to be able to have access to cells from patients with different mutations to test our technology and see whether a similar approach can be used for all the different mutations and be successful, or whether there are only a proportion of mutations that can be targeted that way. I was fortunate enough to go to the world. No, you go, please. Please ask.
Maybe expand on that, because my idea of when you're doing like a cell line, what you're saying is you could essentially make tissue, like muscle tissue, and then you could directly test that?
Yes, so really great question. I'm sorry I didn't clarify earlier, but if we had, for example, a skin cell and a skin cell line from a patient, and often that's the more preferred tissue that a patient or the family members wish to donate, we can still test the technology. We can still test whether the CRISPR-Cas9 technology can correct that mutation, even though it's a skin cell and not a muscle cell, because every cell in our body that has DNA has the same set of DNA, but it's just which cells have different genes switched on or switched off that's different. So we could test the technology in that skin cell, but we couldn't, but then we'd have to try to switch on that gene in skin cells, because it's ACTA1, for example, or the Nemaline's not usually switched on in those cells. But we have a technology that we can use in the laboratory to convert a skin cell into a muscle cell. So yes, we could do that. We could test the technology first in the skin cell and see that it looks like it's working, it's correcting the DNA that's there in that gene, and then we can convert that skin cell into a myoblast cell, and we can see whether the protein that is made, how much of it is the corrected protein and how much is still the mutant protein, for example. So there's actually quite a lot that we can do from a sample from a patient. Particularly for this particular technology, I think it will be very useful. And what I was going to just mention before is that I was at the World Muscle Society Congress. It's an international congress on muscle diseases every year, and I was at that at the beginning of October, and Dr. Ronnie Cohen, who has been working in the CRISPR-Cas9 field now for quite a few years, presented some work on a patient of his that has a dystrophin mutation, and his mutation had never been identified in anyone else in the world. So Ronnie, in his lab, they were able to take his cell line and test the CRISPR-Cas9 technology and show that they could get it to work. And then they actually did go to the time expense and the financial expense of making a mouse model of this particular mutation, and this boy, Ronnie was explaining, had a very rapid progression. So it was a very, very interesting mutation for them to study, and they made the mouse model and they were able to show that it had a similar fast progression. But because they had the cells from the boy to fine-tune their approach, they were able to then take that CRISPR-Cas9 approach and treat the mouse model. And they showed some amazing videos of how this mouse had very little disease compared to when treated, compared to the mouse model that wasn't treated. So that's one way that they were able to go from the patient cells to a mouse model. But what the comment that he made afterwards was really great, saying, for the active disease, there's only one person in the world that's known with that mutation, and that happens a lot with myopathy. In our actin database, we know a lot of, over 200 different mutations, but a lot of those have only ever been found in one patient or one family in the world. And what Ronnie was saying is, what is going to be better? He was asking the question, what will be better? Will it be better for us to test this new technology in the cells from the patients themselves, or in a mouse model that's made with the same mutation? A mouse is a mouse. The DNA of a mouse is different to a human, and we know that humans are very different as well. And a patient might have an actin mutation, but they're also going to have other DNA variations that don't lead to disease, like all of us do. They're going to have some different DNA modification variants. So if you can test the cell line from a patient, perhaps that's the best way to test that approach. And he was saying that he was in discussions, as were others, with the regulatory authorities about what's going to be the way moving forward. If someone can test a CRISPR-Cas9 technology in a cell line from a patient and show that it's useful and works, efficient, are we going to be able to then go straight to treating that patient? Or will we have to make a mouse model, for example, first of all these different mutations? And I think it's very timely, because it's not only for muscle diseases, it's for all different diseases. We need to know what these the next steps are going to be. How difficult is it going to be for us to go from treating a cell line to hopefully being able to test that treatment in a patient? So hopefully that helps answer your question a little bit, May, about how we are going to ideally get into human trials, and we're probably not going to be able to do trials for most of these technologies because there's just not enough patients with the same mutations in the world to be able to do a proper trial.
Thank you.
You're welcome. I'm sorry. I hope I didn't bamboozle you with too much information or too.
No, it was really interesting. I think it's amazing technology.
It is really amazing, and it's very scary. It's very exciting, but it can be very scary in a way as well, because there's the technology exists that we can potentially change any gene in the body, and I guess people are nervous about that. I think us as medical researchers have very pure intent on wanting to be able to cure diseases, and often nasty diseases, but of course the technology is that people can change other genes that do other things as well. So I think it's something that our communities at large need to talk about, and we need to be able to come up with the technologies there, but what are we going to do, where are we going to put the different guidelines for treating patients and humans.
I have a question. Oh, can you hear me?
Yes, I can. Please go
ahead. When some of our children are, of age, can they just volunteer to have a human trial?
That's a really good question, and I'm sorry, I don't know the exact legalities of all that, but my understanding would be so for example, the antisense oligonucleotide trials that have been going on, I think the boys, once they've reached 16, have been able to give their individual consent to participate in those trials. But I've heard clinicians who treat, for example, childhood cancers. They comment on trials in childhood cancers, especially for cancers that are known to be lethal, and they say that a lot of children are enrolled in those trials, and obviously they've been able to get through whatever red tape exists for being able to enroll children into trials. So I'm sorry, I'm not sure the exact legalities of that, but I guess one of the interesting things for us is whether we're going to be able to have a trial or technology that will have an approach that will be able to suit enough patients with myopathy, or whether the approach that we're going to have is going to be an individual approach. Something I didn't talk about today is that one of the approaches that we're hoping with CRISPR-Cas9 may be, like cardiac actin, a one-size-fits-all approach. It might mean that any patient with actin mutations could be applicable, and maybe then would have enough people for a trial. But I guess that's where we have to also be informed as well, we need a trial moving forward, especially with this CRISPR-Cas9 technology. If you can cure someone's or correct someone's cells, is that going to be enough to be able to then do a small test in that person themselves? It's uncharted territory, I'm afraid, at the moment, but hopefully one that's going to get a lot of attention.
Yeah, it is. That's why I was just wondering when someone with myopathy is an adult and they can just choose whether they want to go forward with that or not. But it is that's kind of unethical. But if it's their choice, Yeah,
it's interesting. I think at least in Australia, such a treatment would still have to go through a human ethics committee. So it'd still have to go through a committee at a hospital, for example, or a university, but probably a hospital if we're going to be actually delivering to a patient. And that committee also has to evaluate everything and see whether it's appropriate to go forward, which is obviously a contentious situation. But I think because of the technology, because of how rapidly it's moving, and because of the potential widespread applicability, I'm hoping that these questions are going to be addressed in the near future, and they make it easier for the rest of us with different diseases to be able to move forward.
Yeah, I've been meaning to, my son has some DNA stored at, muscle biopsy at Seattle Children's that I need to get to you, I believe that's what it is.
And Stacey might be able to help out there as well with information about how things work in the States.
Exactly.
Yeah, she emailed me some paperwork. I've just been procrastinating, but this webinar is really giving me some hope, so I'm going to get on that.
Fabulous, fabulous.
Oh, thank you.
You're very welcome. And I can see here that, sorry, you can go. Yeah, it's a great question.
Great question, yeah.
And I think Stacey obviously can talk more about the banking of cell lines, and obviously we're very interested in being able to access cell lines from patients, but it's not just us. I think these cell lines that get banked are then available for any researcher who's looking at Nemaline myopathy. So even if we're not able to do something with that particular mutation, that gene, those cells are then there for future researchers to try other approaches. So I think it's a really valuable thing to do, especially if it's quite non-invasive and just a small amount of skin can be taken, or some blood, for example, and then a cell line made. But Stacey's far more well-versed at this aspect than I am about the logistics of doing this.
Yeah.
And we do have,
thank you.
We do have a great program that anyone on this call, anyone in the affected community can participate in. There's no charge to families at all. It involves a donation of either skin or blood. Most families opt for the blood donation, and that can be paired with a clinical visit that you already have scheduled to do something like check your cholesterol level or vitamin D level. And then what we do is we donate some of that blood to a center, and they develop cell lines that have your exact family's genetic mutation in it, and those are then in a repository where they're available to scientists anywhere in the world to use for their studies. So it's a really great program. I could talk more about it offline with anyone that wants to. Please message me. You can find me on Facebook. I think most of you know how to find me that way. Or email me. I'll post some summarization of what we did today on the webinar, and I'll include my email address in that as well. But it's a really great program. I encourage everyone to participate. It helps the families and it helps the scientists get the material that they need to get the answers that we all would like to have. So if there aren't any other questions, I think we'll wrap things up. Thank you again so much, Dr. Nowak, for your time and this really great presentation about your work and the options that are coming available for treating Nemaline myopathy. And thank you everyone who joined us today.
Yes, thank you. I've really enjoyed the questions, and I'm hopeful. Hopefully we can keep in touch by email and maybe another webinar in the future, and we will keep trying our very best to make some significant advances in treating Nemaline myopathy.